This is the rest of Chapter 1. We're going to talk about classification of microorganisms. Now you have to divide things into different types just to be able to describe them. So that's how we classify them. First we talk about prokaryotes. A prokaryote has no membrane-bound organelles, no nucleus. Pro means before, karyon means nucleus. And these are divided into archaea and bacteria. Now they were just not so very long ago put into one group that was called Monera, but that's out of vogue now. What happened was they were able to realize how different chemically bacteria and archaea are. In fact, I think that one of these is archaea may have more chemicals similar to us than to bacteria, but it just absolutely, they're so different they've got to be divided out. Then eukaryotes, and these eukaryotes, like you and me, you know, like Y-O-U, think about it that way and you can remember it. Animals, plants, they have membrane-bound organelles, and these consist of algae, fungi, protozoans, and animals. And plants, why are plants not on there? Well, we ought to add plants. P-L-A-N-T-S. That's an A right there. Okay, now other viruses, viroids, and prions. Okay, we'll start with prions. A prion is just an infectious protein particle. It's not alive, it was never alive, and you can't kill it. You can burn it, you can autoclave it, it's not going to die. So you don't want to eat something with a prion in it. This is mad cow disease would be an example, and we've got some really fun examples we'll talk about later. Viroids, similar to a virus. Viruses are just a thing of RNA or DNA wrapped in a capsule of some sort. So this may be RNA, and I can't write with these things, oh well, just wrapped in a capsule. And then this will invade whatever body they're going to invade and replicate itself. Let's say if it were a DNA virus and it goes into the organism, it's going to replicate, it's going to copy an RNA off of this and then force the host cell to make copies of the DNA from that RNA. And it's seems a bit complicated, but I'll erase this, it's terrible. They're not alive. Now, there's some thoughts of turning viruses into something of life, but they're not considered alive now. Now, if you're laying there dying of COVID, you're going to say, yeah, they're alive. But as of now, they're not alive. Okay, now, I know some people have a real problem with the E word here, evolution, but we know that bacteria evolve from day to day. In fact, COVID is evolving. It's mutating and changing. And this is talking about ancient times, but COVID was something simple to deal with a little vaccine. Now we have the Delta variant that's breaking through the vaccine. So we may all end up back on lockdown. Oh, what fun. But origin of microorganisms, and generally they look at geological time, and I don't know all those times. The layers of soil and rock are divided into different times, and I don't know all those. There's a lot of fossil evidence of bacterium, prokaryotic life. Most likely microbes were the first thing to exist, and considered to be very old, 3.5 to 4 billion years. Eukaryotic life, as far as 2.2 billion years. that's too far to even get your head around. But where they came from, are back to spontaneous generation, I suppose. But that's for another discussion. We're not talking philosophy or religion or anything in this class. We're just talking what biology says. If you disagree with this, that's fine. Okay, taxonomy is naming stuff. And what kind of stuff? You got to organize and classify it and name them. Now, they're named. Domain is the first big thing. Is divided into the prokaryotes are divided into bacteria and archaea, and then you have the eukaryotes. So that's going to be your domains. Then you have kingdom, phylum. Some books use division for bacteria. Some books even use it for plants. Class, order, family, genus, and species. Now with some, if you're talking about bacteria, you're going to have strains. You have a genus and species of bacteria. You could have a strep that's normal flora in your throat and it doesn't cause any problem. And a different strain, a different variety of it can kill you. Just think about dogs. They're all the same genus and species, but you have poodles and chihuahuas and pit bulls and collies, and so that's what a strain would be. Now, binomial nomenclature is your scientific nomenclature, and binomial means two names and nomenclature means naming, so two name naming. And this was originated by a guy named Linnaeus, and he divided so many things out and named them, and everything now is, we use the same philosophy for naming them. Escherichia coli, since no one can spell Escherichia coli, it's acceptable to just put E. coli. That's an exception to most rules. You could just write E. coli, C-O-L-I. But you've got to have when you write your scientific name out, you have got to have the genus has got to start with an uppercase letter. The species has got to start with a lowercase letter, and it must be italicized. If you're writing by hand, which is not likely, it can be underlined instead. Okay, why do we need binomial nomenclature? I'd just rather say cardinal and bluebird, but you can't really go by that. It doesn't specify what we need. So this is a veterinarian. This is a dog, George, and you're Sally. And she says to you, Sally, will you put George back up in his pen and go get that cat out of the cat room? You say, sure, no problem. What's a cat? Now we see why we need binomial nomenclature. Okay, a Felis catus, I would not mind getting usually, though sometimes I would say they're a little difficult to deal with. Panthera leo is also a cat. I don't think I want to go get him out of his cage. If he's in the cage, he's staying there till I leave the building. You have not described an organism thoroughly if you don't use the genus and species. And you see the uppercase, lowercase, and these are italicized. Kingdoms of life. You have here your eukarya. These are things that are, they have a nucleus, they have organelles. Starts with things like protozoans, Giardia is an example. Other protozoans, flagellates, slime molds, which are pretty cool. Then it goes to animals, fungi, plants, but these are all in the domain eukarya. Now we've got the archaea, and then we got the bacteria. Let me go to the next slide. You will at some point, if you take a standardized biology test, you will be asked to memorize kingdom, phylum, class, order, family, genus, species. They will have them mixed up, and it says which is the correct order. And I can't remember it. without some sort of jingle. This is a pretty popular one. King Philip came over for good spaghetti. I would take a couple of minutes and think that through, because you will probably come across that at some point, and you want to be able to get it right on your test. I've never taken a standardized biology test that didn't have it on there. And all you got to do: kingdom, phylum, class, order, family, genus, species, like we are Homo sapiens. Okay, the two names, genus and species, and I've already mentioned this. The genus is capitalized, the species is in lowercase. After the first use in an article you're writing, in a paper you're writing, you can abbreviate the genus, so E. coli, but you can with other genus and species, just to save space and time. People get tired of reading these long words. Okay, the domain system, Woese-Fox system, divides things into three domains: bacteria, archaea, eukarya, and this one actually has them: protists, fungi, plants, and animals. Microorganisms in health and disease. This will be covered in a lot more detail later, but often deals with biofilms, and I have a section, a slide or two over, I hope, on biofilms. But organisms interact with each other. We have mutualism. They may interact where they both benefit. Like, I have a dog. The dog enjoys the food I give it every day and the dry place to sleep. I enjoy having the dog guard my house and let me know if criminals come in. The dog weighs 15 pounds and has no teeth, but she still will let me know when people come in. She's a very old dog. Commensalism, one organism benefits, the other doesn't care one way or the other, such as fish that swim along beside the big shark. The shark can't see them, but they benefit because they're protected by that shark, actually, if they stay close enough to him. And when he eats, they can get bites of food that fall off. Synergism, the two things work together so that they actually work better than they would have alone. Parasitism, and this is the big thing we talk about in disease. One organism hurts another. You get worms, you are being parasitized. There's a lot of normal flora, normal organisms growing on your body, in your body. There's so many things that are growing there. In fact, if you do take antibiotics that get rid of the normal flora in your gut, you probably will get sick. A pathogen is a bad guy bug, things that make you sick when you are infected with them. And this is just a couple of little things here. Foodborne illnesses. You may be infected with the actual organism, or you may just have a toxin, like in botulism. You don't have the infection of botulism. Generally, you just have the toxin, and if somebody can keep you alive on a vent until you get over it, you'll live. Waterborne diseases come from contaminated water, fecal contamination, and Giardia is an interesting one. Airborne diseases, COVID-19. You go in a bar, you breathe, you get COVID-19 unless you take a vaccine. A couple of things in applied microbiology. Food production. Yogurt and bread, you use yeast, which is fungus, to make bread. And you use lactobacillus to make yogurt. That's a bacteria. Alcoholic beverages, I believe those are fungi. Water treatment. They pour this, turds and water into these big ponds, and they aerate them for a long time, and with air flowing, and it changes the bacterial organisms until they're no longer a problem. Now, pharmaceutical agents, they make penicillin out of organisms. Monoclonal antibodies are made out of a chimera. A chimera is a combination, an organism that has two types of DNA. And one of the more popular monoclonal antibodies is used, say for an MS, I think it's called Rituxan, is made, they take human lymphocyte DNA and they mix it with the ovaries, the cells of the ovaries of a Chinese hamster. I don't know what a Chinese hamster looks like. See, we need the genus and species name. And they put food in and they put out the $15,000 drug. Now, this is the chimera. And it happens as an accident of nature in plants. And it's really cool when it does. We can have monoclonal antibodies which are genetically engineered. This is actually was one of my plants. I think I somewhat killed it after a year or two, but it's a chimera. This is an African violet. Now the leaves don't indicate that it's a chimera, but see these flowers with this is very, very abnormal. And I had forgotten it was a chimera, and when I replanted it, see all these leaves fell off? I planted it in a big terrarium or small terrarium thing, and look, I got this kind of plant, and I got this kind of plant. And somewhere in there, eventually, I had one chimera. But if you take the chimera and just root the leaves, you're going to get the DNA of one plant and these white ones, they didn't have any chlorophyll, so they didn't do so well. The green ones did much better. If they had a little green, they could live. You got to have the chlorophyll to make it, though. Okay, in agriculture, we talk about soil microbes. Plants have to have them to exist. Nitrogen is pulled out of the air to make it usable for plants by bacteria. Bioremediation, there are actually petroleum digesting bacteria. Fuel, ethanol is in your gasoline, and they take corn and they basically brew it down into corn whiskey and distill it and put it in the alcohol. It's supposed to help the environment. It helps the corn farmer, but I'm a little concerned that it probably takes more energy to produce this ethanol than it does than you get out of it. Now, forensic science. There was a case, we'll look at criminal justice and we'll look at these others later. A little girl's body was found and just abandoned, and the forensic scientists took bacteria from her intestinal tract, and they knew from the bacterial flora that she did not come from that area, and they figured out, oh, she came from a certain other area of the United States, because apparently someone has in a computer base what your gut bacteria looks like here and there. So then they got on the file system and looked for missing children, and they found one that fit the description. Turns out the parents were arrested and imprisoned. They had killed the child and abandoned her in another state. Bioterrorism, when you release an organism or a virus on people to make them sick and die. And that's all we'll talk about on this slide. Thank you very much.